discussing chapter number 9, biomolecules. We have discussed many biomolecules, and in the last session we were discussing a biomolecule called as enzymes. Enzymes are acting as a biological catalyst. They increases the rate of reaction. They speed up the velocity of reactions, and thus they are useful in doing a biochemical reactions in our body. Without the enzyme, the chemical reactions in the body that slows down and we are not able to survive at such slow speed. Hence there is a need of an enzyme. So that we have discussed yesterday. We have also discussed that how the enzyme increases the rate of reaction. Okay, by lowering the activation energy, the enzymes increases the rate of reactions. Apart from that, we have also seen the nature of enzyme actions. Means what is a mechanism by which the enzyme is capable of increasing the rate of reaction. So that we have already discussed in the previous session. Now today we have to learn the factors which affect the enzyme activity. So let's learn the factors which affect the enzyme activity. So here we are going to start with factors affecting enzyme activity. Factors affecting enzyme activity. You know that the enzymes, most of the enzymes are made up of proteins, and the protein might have a primary structure, secondary structure, tertiary structure, or the quaternary structure. Here in most of the enzymes, the tertiary structure of protein may exist, and you know that in the tertiary structure there is a three-dimensional arrangement of single polypeptide chain that leads to a formation of active site. Active site, you know the meaning of active site. Active site is the services or the pockets which are going to be formed. Okay, and this is necessary for the activity of enzyme. Remember, the active site is necessary for the activity of an enzyme. But any factor that alter the active site or they alter the tertiary structure of a protein, they affect the enzyme activity. Anything which can affect, or any factor which can affect the tertiary structure of a protein or the active site, they affect the enzyme activity, and they are called as factors affecting enzyme activity. So let's first start with one of the important factors. So one important factor is temperature. Second factor which affects the enzyme activity is the pH. The third factor that affects the enzyme activity is concentration of substrate. Clear? Similarly, the concentration of inhibitors, or you can say that the concentration of enzymes, they all are the factors. But according to the book, we just have to learn the three factors: temperature, pH, and the concentration of substrate. But apart from that, the many factors are that which can affect the enzymatic activity. Like for an example, that is a concentration of enzyme, concentration of inhibitors, the metals and the cofactors which are present over there, they can also affect the enzymatic activity. But we will learn these three factors which are affecting the enzymatic activity in detail. Okay? So let's start with the first factor, what we call it as temperature. Okay, the temperature is the first factor which can affect the activity of an enzyme. Okay, you know that the enzymes, they are going to be active in a narrow temperature range. Every enzyme shows its maximum activity at a specific temperature, and this temperature is called as optimum temperature. So first of all, I will tell you what is the optimum temperature. Optimum temperature. What is an optimum temperature? The temperature at which enzyme shows its maximum activity. Maximum. Activity. This is called as optimum temperature. What it is called as? Optimum temperature. What is optimum temperature? The temperature at which the enzyme shows its maximum activity. This is called as an optimum temperature. So if you plot a graph of enzyme activity, versus temperature. Clear? Versus temperature. I am writing over here. I am writing over here scale 0 degree, 10 degree, 20, 30, 40, 50, 60, 70 and so on. This is the graph of enzymatic activity or the temperature. So usually this is a temperature. You can say that between the 30 degrees Celsius and the 40 degrees Celsius. Okay, here I have shown the temperature in the degrees Celsius. At the temperature between a 30 and 40 degrees Celsius, the enzyme shows the maximum activity. Suppose assume that this is the maximum activity. Below this temperature, below this 30 degrees Celsius, there is a decrease in the enzymatic activity, and above the temperature, above the 40 degrees Celsius also, the activity of enzyme gets decreased. So this temperature is called as optimum temperature. What it is called as? Optimum temperature. Okay, usually for most of the enzyme, the optimum temperature that lies in the range of 30 degrees Celsius to 40 degrees Celsius. This is a range in which they shows the maximum activity, and this temperature is called as optimum temperature. Any deviation from this temperature, deviation means either lower temperature or higher temperature, lower than 30 degrees Celsius or the higher than 40 degrees Celsius, they decreases the enzyme activity. Because what happens when the low temperature provides, at the low temperature the enzyme becomes inactive. I again repeat, at the low temperature the enzyme becomes inactive, and at the high temperature the enzyme structure gets a denatured, because we have already learned in the previous session and the last session that when a high temperature is provided, at the high temperature the tertiary structure of a protein gets denatured, degraded, and once the structure of enzyme gets denatured, it is not able to perform its function. That means it cannot speed up the rate of reaction, or it cannot increases the velocity of reaction. That is going to happen. Okay, so remember, at low temperature, lower than the optimum temperature, the enzyme becomes inactive. At a very high temperature, the enzyme gets denatured. So the enzyme activity gets decreased in high and the low temperature both. But the maximum activity you can show it at optimum temperature. Usually, where the temperature is going to be increased by 10 degrees Celsius. Usually remember, when the temperature is increased by 10 degrees Celsius, suppose if you assume that this is the activity of an enzyme at 10 degrees Celsius. Okay, whatever it may be. If you increase this by 10 degrees, so 10 plus 10, it becomes a 20. So at every 10 degrees Celsius rise, the activity of enzyme usually gets doubled. It gets a double. So from 10 to 20, at 20 degrees Celsius, the activity of enzyme is double than the activity which occurs at 10 degrees. Same happens with 30, same happens with 40. But now when it goes beyond a 50, the activity decreases the half. The activity decreases the half. So suppose assume that this is the activity at 40 degrees Celsius. Okay, and this is the activity at 50 degrees Celsius. So if you compare this, the activity at 40 degrees Celsius and compare the enzyme activity at 50 degrees Celsius, the enzyme activity at 50 degrees Celsius is half than the activity which occurs at the 40 degrees Celsius. Clear? So every 10 degrees rise, either double the rate of reaction or half the rate of reaction, because if there is a below 30 degree, then double the rate of reaction. If beyond the 40 degrees Celsius, it reduces the rate of reaction by half. Okay, so this is the effect of temperature on enzyme activity. In short, you just have to remember that the every enzyme shows its activity, maximum activity at optimum temperature. If any deviation occurs from it reduces the enzyme activity. So this is called as the effect of temperature. Okay, most of the enzymes, they shows the optimum temperature in the range of 30 to 40 degrees Celsius. But I have already told you few exceptions in the previous sessions. The exceptions are the enzymes which are isolated from the organisms that you can find in the hot springs, vent, and the sulfur spring. These are the locations from which, if you isolate a bacteria, the enzymes of this bacteria can also survive at higher temperature. Okay, they can also survive at 70 degrees Celsius, 80 degrees Celsius, and so its activity. Clear? But they are exception. Usually the optimum temperature is in the range of 30 degrees Celsius to 40 degrees Celsius. So that is about the first factor that affect the enzyme activity. Same thing happens with pH. But before I start the pH, I will also tell you what is the shape of this graph. The enzyme activity versus temperature. What is the shape of a graph? It is a bell-shaped curve is going to be obtained. Which shape? Bell-shaped. Okay. Bell-shaped curve is going to be obtained. Enzyme activity versus a temperature. Okay. Next, now we are starting with another factor. Okay. And here the another factor is pH. pH means it is a negative logarithm of molar concentration of a hydrogen. This is called as a pH, minus log H plus. You might know the equation, pH is equal to minus log H plus. So it determines the molar concentration of a hydrogen which is present in it. Okay. Now suppose assume that this is a 7 pH. You know that the pH that is ranging from 0 to 14. The 0 is the lowest pH, 14 is the highest pH. So on the x-axis I am drawing the pH. On the y-axis, I am taking the enzyme activity. Okay. 0 is the lowest pH, 14 is the highest pH. 7. This is a 7, it's considered as a neutral. I hope you know that. The pH between 0 to 7, it's called as acidic. The pH between 7 to 14, it's considered as basic. So this is an acidic pH. And this one is a basic pH. Okay, this is a range which is for basic pH, this is a range for acidic pH, and the 7th is neutral. Now, similarly, like a temperature, the enzyme also shows its optimum activity at optimum pH. Here the optimum temperature, now we are taking the optimum pH. What is the optimum pH? Optimum pH means the pH at which the enzyme shows the activity. Okay, or the maximum activity. I have to modify it. The pH at which the enzyme shows its maximum activity is called as an optimum pH. Some enzyme shows its optimum activity or the maximum activity at acidic pH. Some enzymes shows its maximum activity at a basic pH. Like take an example of pepsin. You know the example. Pepsin is an enzyme that is released in the gastric juice and that is responsible for digestion of a protein. This pepsin, they shows its maximum activity at acidic pH. Okay, that's why the environment of a stomach is acidic. Okay, if it is not acidic, then the enzyme doesn't become active. Pepsinogen doesn't get converted to a pepsin. You already know it. You might have studied in the 10th standard that pepsinogen converts to a pepsin in the presence of dilute hydrochloric acid. Dilute hydrochloric acid provides a acidic pH, and at this pH, the enzyme shows its a maximum activity or the optimum activity. So some enzyme shows the maximum activity at a acidic pH. Some enzymes are showing the maximum activity at a basic pH. So let's see. If you see that pepsin, if you take, then it shows the maximum activity around the pH like this, 2. Okay, so this is a pH 2. I say. Below this pH, below the 2, the activity gets decreased. Even above the 2, the activity also gets decreased. So this is an optimum pH. Optimum pH for which enzyme? Here the optimum pH of pepsin. This is one example. Okay. Similarly, I have also told you that some enzymes are also active at basic pH. So, like for an example, take an example of trypsin. Instead of pepsin, we are taking the trypsin. So this is an optimum pH of trypsin. Okay, at which it shows the maximum activity. Below this pH and above this pH, activity gets decreased or the enzymatic activity gets decreased. So similarly like temperature, here the shape of a graph is, like for a pepsin also the graph is bell shaped, for a trypsin also bell shaped. Okay, so here also a bell shaped graph is obtained. Ultimately you also have to remember that as the deviation in the pH occurs from the optimum pH, there is a decrease in the enzymatic activity and the enzyme shows its maximum activity at optimum pH. For some enzyme, the optimum pH is acidic. For some enzyme or the other category of enzymes, the optimum pH is in a basic range. Okay, so thus you can get a bell-shaped curve. The enzyme activity versus a pH graph. Okay. Now the third factor which affect the enzyme activity is a concentration of substrate. Okay, this is a concentration of substrate. This is a third factor which affect the enzymatic activity. So let's discuss the third factor which affect the enzymatic activity. You know the basic reactions. What happens? E plus S, E means enzyme, S means substrate. They leads to a formation of ES, what we call it as enzyme substrate complex, which leads to a formation of EP, enzyme product complex. And ultimately, finally, what is going to be formed? A product and enzyme gets liberated. Okay, they get separated. The enzyme can be utilized to catalyze one more substrate. This is what happens. But you know that this substrate that binds with an active site of an enzyme, and the binding of substrate and enzyme is like a lock and key. Like a lock and key. Like a particular key only open a specific lock only. Okay, it is not like that a key can open any lock. Okay, because there is a matching of the grooves. If there is a matching of a groove, then and then it can open the lock. Like suppose for an example, it is like this is a lock and this is a key. So it can only goes like this. It can only goes if it matches and can open it. If it is not matching, it cannot open the lock. कदाच key अंदर जती रहे, पण lock तो open ना थाय, ज्यां सुधी एना grooves match ना थाय त्यां सुधी. Similarly, the binding of enzyme and the substrate is also like a lock and key, Okay, so the substrate can bind to a specific enzyme only. It cannot bind to any enzyme. Like the maltose can bind with a maltase, or maltose is hydrolyzed by maltase. It is not hydrolyzed by the sucrase or lactase. Okay. Similarly, the sucrose is hydrolyzed by sucrase, not by the pepsin or trypsin. Clear? Because it's working like a lock and key. There is a specific enzyme for a specific substrate only. Okay. So same thing happens. I will show you with a diagram. Like for an example, this is an active site which is present in the enzyme. This is an enzyme. Assume that it is an enzyme. This one is an active site. Okay. This is an active site. Okay. I am showing you. This is an active site. Now assume that this is a substrate. Yes, this is a substrate. So now the enzyme and the substrates, their grooves are matching. So they work like lock and key, and then they can combine. They can combine. Okay. Let's see how the combination takes place. Clear? It can combine. This is a substrate and this is an enzyme, and that leads to a formation of enzyme substrate complex. बराबर से जो आप मैचिंग तत्व से तो enzyme substrate complex बनते हैं, ultimately it leads to a formation of product. But suppose assume that if this is a substrate, like this is a substrate, can it combine with this enzyme? Suppose assume that this is a substrate 1. For easy differentiation, I am giving this as a S and this is as a S1. So the S can combine with this E and form ES, but this S1, it has a different shape. So it cannot fit with this groove and cannot form the enzyme substrate complex. If it cannot, then the product is not going to be formed. Okay. So before learning this, the effect of concentration of substrate on the enzyme activity, you should know that how the enzyme works. The enzyme works like a lock and key model. Okay. If the substrate and enzyme fits, then and then the product is going to be formed. Otherwise, it is not going to be formed. Okay. So there is one thing is clear now, that a specific enzyme can only catalyze a specific substrate only. It cannot catalyze any other substrate. Like for a maltose, the enzyme is maltase, not any other. Okay. Clear? So this is what happens in the case of a working of an enzymes. Now let's discuss how the concentration of enzyme act, or how the concentration of substrate will going to act. Now I will also tell you one example. Like for an example, this is a bus. I am showing you, this is a bus. Okay, very small bus. It has just 5 seats. Or suppose assume that this is a car. It has 5 seats in it. Okay, assume that this is a 5 seats this is a car which you can denote it as an enzyme. Now there are people. Okay, they are the people. You call it them as a substrate. This is for just an example, for easy understanding. This is assume that this is a car. Now it is a 5-seated car and there are 2 people are present. Okay, here we are comparing the car as an enzyme and the people we are comparing it as a substrate. Now what happens? You know the function of this car. This car carry the people from one place to another. They help in the transportation of people from one place to another. Now these people want to go. Can they sit over here? Yes, they can sit over here. Okay, and they are transported from one place to another place in a single run. Now I increase the people. Instead of 2, I am taking it 3. Okay, so in the first case, only 2 people are going at a place. Now I am increasing the concentration. I am increasing the number of people. That means I am increasing the number of concentration. Okay, or the concentration of substrate. Okay, because here we are taking the people as a substrate. So can it carry 3 people at a time? Yes, it can carry 3 people at a time. So in a single run, the 3 people can carry. Now I increase this to 5 people directly. Instead of going to 4, you know that even it can also carry a 4. Now I take a 5. So the car can carry also a 5 people at a time because there are 5 seats. But now assume that there are 6 people. So can a car carry 6 people legally? No. At a time it can carry how much people? At a time it can carry 5 people. Okay, it can carry one more people which has been left, but in the second run, okay, in the first run it can only carry a maximum 5 people because it has a 5 seats. Similarly, okay, so as you increases the concentration of substrate, I am drawing over here. Okay, this is enzyme velocity or enzyme activity. And here the x-axis is concentration of substrate. So 2 થી 3 કર્યા, 3 થી 4 કર્યા, 4 થી 5 કર્યા, ત્યાં સુધી કોઈ વાંધો નતો. They are carrying the people at a one time. But જેવું 5 થી 6 થયું એટલે they have to go for 2 runs. Okay, બરાબર છે, 2 ધક્કા ખાવા પડશે. The same happens over here. Okay, so initially as you increases the concentration of substrate, the rate of reaction gets increases, but after the saturation it becomes a stable, and this is called as maximum activity or maximum velocity, what we call it as Vmax. That is going to be achieved. Okay, clear? So initially the rate of reaction increases, then it becomes a stable, okay, or it becomes a saturate. Okay, and thus there is a effect of concentration of substrate on the enzymatic activity. Clear? So I hope so it is clear. There is a effect of concentration of enzyme. Now see the another thing. Now what happens? Now it is clear to everyone that substrate binds to a specific enzyme if it matches, because it's like a lock and key. So this substrate can bind with the enzyme, but this substrate cannot bind with this enzyme. Okay, so if this S, then it can be catalyzed. If S1, then it cannot be catalyzed. But there are also very smart people and the chemicals. Like for example, see, if this is a kind of a structure I have, what I call it as S2. This is a kind of a structure. Now you know that what is a kind of a structure of enzyme? This is a kind of a structure of an enzyme. What we have drawn over here. This is just for your easy understanding. This is an enzyme. Clear? Now, does it fit in? The structure of S and S2 are not same. They are different, like you can see. There is a difference over here. Okay? They are not same. Actually, the enzyme is meant for this. Okay? Enzyme is meant for this S. But now it has similar looking structure. The structure of S and S2 are similar looking. The structure of S and S1, they are completely different. But the S and S2, they are similar looking. Now what happens in this case? Here with this enzyme, the S can also bind, even the S2 can bind because they are matching, their grooves are matching. So what happens? The S and the S2, they compete with each other to bind with this site. બરાબર છે? લાઈક માની લો કે અત્યારે તમારી સામે બેન્ચીસ પર બે સીટ છે, બે છોકરા બી છે, બે સ્ટુડન્ટ્સ બી છે. હવે ત્રીજો સ્ટુડન્ટ આવ્યો, એ કહે છે કે મારે અહીંયા શું છે, તો શું કરશે? બે જણા ઝઘડશે, they compete with each other. બરાબર છે? અને તમને ખબર છે કોણ વિન થશે? Those who has a higher power, જેની જોડે તાકાત વધારે હશે એ જીતી જશે. Same thing happens over here. The substrate and S2, they compete with each other because the enzyme is same. Clear? Who will win? Those who has a high affinity or those who has a high binding capacity. જે ઝડપથી બાઇન્ડ થઈ જશે, જે સ્ટ્રોંગલી બાઇન્ડ થશે, that will win. Okay? But this S2 બેમાંથી એક જ જગ્યા લઈ શકે, ક્યાં S ક્યાં S2. But this S2, once it is occupied, it prevent the binding of S. And if S cannot combine, the product cannot be formed. Clear? So here, this S2 we are calling it as I. Okay? This is called as inhibitor. What we call it as? Inhibitor. So what is an inhibitor? Inhibitor I, it prevents a binding of S with an enzyme. And once the inhibitor binds with an enzyme, it cannot catalyze the reaction. A reaction આગળ થશે જ નહીં because S binded નહીં થયું, ES complex બનશે જ નહીં, and the product is not going to be formed. Clear? So this is what going to be occur in the case of an inhibitor. Okay? And this kind of inhibition is called as competitive inhibitions because both are competing with each other. Okay? There is also another kind of a example. Like I have told you one example that 3 students are there. બરાબર છે? And there are only 2 seats. બરાબર છે? તો they fight with each other. જે વધારે તાકાતવાળું હશે જીતી જશે. પણ એ તો 3 સ્ટુડન્ટ હતા એટલે ઝઘડતા હતા. But now suppose assume that 3 સ્ટુડન્ટ છે અને હું છું સર, જગ્યા તો બે જ છે. ક્લિયર? તો સ્ટુડન્ટ હશે તો એ ઊભા થઈ જશે ને એની જગ્યા કોણ બેસી જશે? ટીચર. બરાબર છે? તો ત્યાં ફાઇટિંગ નહીં થાય. Here there is no competition. So this kind of inhibition is also called as non-competitive inhibitions. But then you don't have to learn. You just have to learn the competitive inhibition. Competitive inhibitions, that means here there is a structural similarity between the substrate and inhibitor. It occurs because structural similarity. Like substrate and inhibitor has structural similarity. They are mostly similar. So they compete with each other to bind with the same enzyme. And so that's why they affect the enzymatic activity. So that's all about the factors affecting the enzyme activity. I will tell you the example of these inhibitions. Like usually, take the example. This is an enzyme, Succinate Dehydrogenase. This is the name of enzyme, Succinate Dehydrogenase. For this Succinate Dehydrogenase, the substrate is succinate. Succinate is a substrate. So usually succinate is going to be converted into some other moiety. બરાબર છે? Clear? But a similar looking structure like a succinate is a mevalonate. If you see the structure of succinate and mevalonate, they are same. Actually, succinate binds with this enzyme. Okay? પણ એ જગ્યાએ હવે બે છે કે બે સરખા જેવા સ્ટ્રક્ચર છે. mevalonate and succinate has similar type of a structure. Not exactly same, but the similar looking structures. So the mevalonate and succinate will compete with each other because there is only one enzyme. Rather, they compete with each other to bind with the same enzyme because for both of them the same enzyme. JP bind થશે એ પ્રમાણે આગળ રિએક્શન થશે. Succinate bind થશે તો આગળ થશે, mevalonate થશે તો બીજું કંઈક બનશે. Clear? A product is not going to be formed. What is desired? Okay? So that's all about the factors affecting the enzymatic activity. Remember, these are the 3 factors which affect the enzyme activity: temperature, pH, and the third one is concentration of substrate. Students, learn it properly. This is an important short note regarding the examination point of view, so learn it thoroughly. Students, keep learning, keep watching, and like and subscribe this channel. Thank you for watching.